Okay, so we'll move right on to science stuff. Any questions come up about just sort of process administration, anything that you've seen there? Okay, good, silent group. I'm going to talk first about signaling in neurons, and I'm doing it principally from the perspective of trying to help understand what it is about the way neurons work that makes them visible to our imaging technologies. Obviously, the emphasis on this course is on functional MRI. So some of this, maybe most of it will be review for you, but maybe the perspective will be a little different. So we're going to talk about sort of the basics of neurons, how they communicate, and what are some of the energetics involved in this, and also a little bit about how information is encoded. And hopefully I'll say a few words about MRI and EEG, which are two different views of the same activity. So here are some lovely camera-lucided drawings of a number of different neurons, the number being 5. 5 is a tiny sample, of course. The representative number, it was more like trillions. But neurons are very striking cells in the body, in part because they're incredibly large, many of them. A pyramidal cell like this one might be millimeters long, just in the portion of it that sits right inside the brain. Pyramidal cells are probably the most ubiquitous cells inside human cortex. They're large, they're easy to see, and they have enormous amounts of interesting function to them. Some cells tend to be what we call stellate. They have a sort of a spherical shape to them. Whereas the pyramidal cells, as you can see, have this odd sort of very polarized shape. The cell body's down here. The so-called dendrites, which are the incoming information, is all on one side of that, whereas the stellate cells have the sort of dendrites all the way around them. They capture information from a more spherical area. Many cells have this sort of bipolar configuration to them, and they're somehow integrating kind of, well, in this case up and down, but usually left and right. These gorgeous hedgerow cells, Purkinje cells in the cerebellum, are very, very striking in that they're almost planar, and yet they have this dramatic arbor in the orthogonal plane to that. What distinguishes neurons from other cells are several morphological features. One is that they have a single axon that comes out of the cell body. And the axon is understood to be the output transmission from neurons. And they have typically many, many dendrites, finger-like things out there from which they capture information. And they are also electrically excitable. Like all cells in the body, they have a potential difference, a voltage potential difference between inside and outside. The difference is that neurons can change them. So if you were to stick an electrode inside a neuron, which can be done, you can measure a voltage difference between the inside and the outside, and the neurons are typically about 1/10 of a volt lower, tenth of a volt potential difference negatively compared to the outside of the brain. And so the first thing I want to talk about is how that potential difference takes place. Here is we can imagine the inside of the neuron on the right and the outside of the neuron on the left, and inside of it are solvated ions, that is salts. So positive charge separating in the atoms from the negative charge, and some of it freely diffusing around. If you have a barrier between these two, what you'll find is that there's essentially the same number of positive and negative charges on both sides of that barrier, because it's actually quite hard to remove charge from a chemical solution. On the other hand, you can see there's a large concentration difference here. There's kind of more salt inside than outside from here. Were I to open that barrier, what will happen is that the charges will move down their concentration gradient, right? Much as if I opened a container of perfume right over here, that not too long from now, people in the back row would smell it. Things go down their concentration gradients. It's a very, very high-powered statistical function. The improbability of having everything in one place is too high, and things move to their more probable location, which is an even distribution. And they move enough that some of them will cross onto the other side of the membrane. Now you can see the membrane was designed, let me back a slide. The membrane was designed in such a way that only some of the charges are able to move across, the small ones, in this case the positive charges. And that makes the left-hand side of this thing, the extracellular space, more positive than the inside space. And that's how you generate the potential difference between the insides. It's relatively easy to calculate the voltage difference that happens as a function of concentration. This is the so-called Nernst potential. There are many charged ions, many different kinds of charged ions, and some of them can diffuse freely between the inside and the outside of the neurons, and some cannot. But principally, the positive charges are the positive potassium charges are in very high concentration inside the neuron and very low concentration outside, and the membrane is permeable to these, which ends up driving positive charges out, makes the inside of the neuron, as they say, about 1/10 of a volt negative. Other charges are also out of equilibrium. For example, sodium is roughly 10 times more concentrated outside than inside. However, the sodium ion can't get through the membrane, and so those concentration differences are sustained. Again, you can calculate this Nernst potential thing for multiple different numbers of mobile charges when you take into account the concentration outside and inside, as well as the permeability of the membrane to those. So if the membrane becomes more permeable, obviously the charges move down their concentration gradients. The cell membrane itself is a lipid bilayer, just a little micelle of fat, and it is constructed in such a way that the polarized head so fat is made up of a polar head, something that's charged, and a non-polar tail, basically a hydrocarbon tail. And when you throw this stuff all together and you shake it, what happens is that the fat tends to form bilayers. So you shake it in water, I should say. The fat tends to form bilayers in which the polar portions of it have high affinity for water, which itself is a polar molecule. And the non-polar tails have high affinity for each other, so they make this layer. And because the inside is made of fat, water doesn't go there. And so these end up being a pretty powerful barrier to charged particles, because charged things don't go through them. The membranes are impossibly thin at 5 nanometers, and yet there's on the order of 100 millivolts separating the two, which means there's a voltage gradient of 10 million volts per meter, an extraordinarily high voltage gradient, which, as you may know, means that there's an enormous amount of force. The charges are pushing at each other for this, and it's astonishing that the neurons can sustain that, or that any cell can sustain that. So one of the most interesting features of neurons is their electrical excitability. Here is a drawing of an experiment in which this is the axon, the output fiber of a neuron, and somebody has placed into it a current source, a source of electrical charge. And you can just push current into the neuron. And if you were to push, for example, positive charge into the neuron, what happens is that the inside becomes more positive compared to the outside. If you push negative charge in, the opposite happens. The inside becomes more negative. And you can measure that voltage difference with another electrode in there. So here's some data from experiments. If you take a small amount of charge, negative charge, into the neuron, what will happen is the neuron will respond by going more negative. Okay, so this is the potential measured inside the neuron. It takes a little time to change, which I'll talk about in a minute. And then you turn the current off and it goes back to its resting potential. If you put twice as much charge in, the neuron becomes twice as negative, behaves effectively linearly in that manner. If you put a small amount of positive charge in, a similar thing happens. The neuron depolarizes slightly. And if you increase the amount of charge, something else happens, which is that the neurons at some threshold become non-linear. They produce a single peak of a fixed duration and shape, which is more or less unrelated to the shape of the charge, the time course of the charge coming in. If I put in still more charge, I get the same response out. It's stereotyped. I mean, this so-called action potential. A lot is known about the action potential. Hodgkin and Huxley, who are two geniuses for which they got the Nobel Prize, worked this all out in some great detail back in the early 50s. What they showed is that if you depolarize the neuron enough, what happens is that all of a sudden the neuron becomes permeable to sodium. The sodium, which is in high concentration outside, comes rushing inside and makes the neuron more positive. And just about as quickly, the potassium current goes up, which now is going to push the neuron the other way, right, towards more negative. And for reasons that, that are not entirely well known, the sodium permeability is transient. It just stays open for a small amount of time, and then those gates close. And the combination of the incoming positive charge and the outgoing charge through potassium charge ends up producing this brief action potential. So given that that's what goes on, let's kind of think about the energetics of this thing for a minute. Every time that a neuron fires, sodium comes into the cell. And we know that, a typical cell has something like 10 to the minus 12th liters of fluid in it. You can calculate that just from more or less the size of the cell. And if you have 40 millimolar sodium in there, you can actually calculate the number of sodium ions that must be in here, which is about 10 to the minus 14th mole, which is about actually 10 to the minus 14th ions. Right? I'm sorry, 10 to the 14th ions, excuse me. Get that right. The action potential changes the neuron from about minus 75 volts to about plus 55 millivolts, which is a change of about 0.13 volts. And you can calculate how much charge had to move based on the fact that the neuron has this membrane, which acts like a capacitor. A capacitor is simply an electronic device which separates charge by having an insulator, and the membrane is an insulator in here. In order to change the voltage across a capacitor, you have to put more charge in. And so you can go ahead and calculate how much charge had to move into the neurons in order to get that action potential. That's about 10 to the minus 12 moles. Given that, so this is 10 to the minus 12 moles per square centimeter. So given that the neurons are about 10 to the minus 5 square centimeters of surface area, you're moving about 4 times 10 to the minus 17 moles of sodium, which means that the concentration of sodium is increased by about 0.1% with each action potential, which means that if you fired an action potential 1,000 times, the neuron would be depleted. And people have actually verified these kinds of numbers experimentally, which is kind of neat. So what keeps the cell from being depleted? It's a little battery. How do you charge it? And you charge that battery with something known as the Sodium-Potassium Pump, which basically is a machine on the surface membrane of the neurons, which burns ATP, adenosine triphosphate, and makes a mechanical motion that takes sodium ions from the inside and pulls them out, and at the same time, or at a separate part of the cycle, like a two-stroke engine in a lawnmower, it also lets potassium ions in. And this, the amount of energy involved in this is really quite extraordinary, since we're now taking charged particles and moving them through a 10 million volt per meter field, right? An enormous amount of force in order to do this, but it does this by more or less exploding ATP. I guess I'll refer to this in a little bit, but this takes an enormous amount of energy, right? And you're burning sugars to make ATP, and then you're burning the ATP, and the neurons demand a lot of energy. And they need that energy to recover their resting state potential with each action potential. There's some other interesting properties of neurons. this is a sort of an electronic diagram. How many people recognize what this device is? Just raise your hand so I can see it. A few, okay. This is a device called an electrical resistor. It resists the flow of current. This is an electrical device called a capacitor. I talked about them before. And you can model the membrane, which is this gray area here, as a combination of resistors and capacitors. And in fact, you could build a device like this with your own two hands, making it out of resistors and capacitors, or you can measure these kinds of effects. But if you were to apply a voltage difference between inside and outside, just at a point over here on the neuron, what you'd find is that if you measure the potential difference at different distances away from that point, it falls off. And that's because of the leakiness of the membrane through these resistors. Okay? So here, just where I insert the current, I get whatever is my applied, just where I insert the voltage, whatever is my applied voltage. And as I go away, it falls off. And it falls off exponentially with a spatial constant, lambda, determined by the resistors, which is, boy, I'm missing a number on here. Lambda is on the order of microns, of tenths of a millimeter to millimeters. Okay? It's pretty small, which means if you move away a couple of millimeters from wherever you're inserting the voltage, that there's virtually no potential difference created by that. That has interesting effects, which have to do with how the action potential is created and transmitted. Another feature from this is because there is a capacitance in here, that adds a time constant to the neurons. It takes time to bring charge to a capacitor. So if you apply instantaneously a step in current over here, the voltage takes a while to settle in. And the time constants for neurons is in the order of milliseconds. Okay? Relatively long. So neurons communicate with each other by making these action potentials, and then the action potentials get transmitted down the axon and where they reach other neurons. But how they get transmitted down the axon turns out to be, it's very, very well known, but it seems to be a source of confusion among many people who are in the functional neuroimaging business. The axons are not wires. It's not that they conduct charge through the inside of the axon the way a copper wire does. In fact, what actually has to happen is different. You have an action potential forming, say, right over here. And what happens is that if you measure at a small distance away from that point, the amplitude is lower because of the spatial constants of the neurons and slightly delayed because of the time constants of the neurons. And if a neuron, if the voltage is below threshold for the sodium channels to open, it doesn't form an action potential. However, immediately adjacent to my action potential over here, it looks like position V2 is going to form an action potential because it's crossed its threshold. So the neuron reforms the action potential, and then everybody else down the line starts to see a higher voltage, and the action potential propagates through the neurons. So this process of propagation to the neurons is, number one, not instantaneous. Number two, it's not like a piece of wire. The amount of current that's flowing through the neurons this way along them is very, very tiny. Most of the current flows across them. It's relatively slow because of the time constants of the neurons. So the typical velocity is only in the order of meters per second. So the distance from my brain to my knees is on the order of a meter. And my brain has to somehow communicate to my knees in time for me to move them out of the way when somebody's going to kick me. And so something has to happen over here because this is a slow process. What happens is called myelin. Myelin is actually just more cell membrane, just like I showed you before, lipid bilayers, but wrapped around itself in these large spirals. So this is an EM, a cross-section through an axon, electron microscope cross-section through the axon. And you can see actually the polar head and the non-polar tails, polar heads, non-polar tails, and so on for the membrane wrapped around itself. And because this stuff is impermeable to charged particles, what it does is increase the effective distance between inside and outside the neuron. What that does is change the capacitance of the neuron. It actually lowers it, and it also increases the resistance to current going through it, both of which have the effect of increasing the space constant on the axons and also reducing the time constant. Here's another gorgeous picture of scanning EM through myelin. And you can see the axons and the lipid layers. If you study the neurons up close with a microscope, you'll find that these myelin layers are in little segments, each of which is produced by some little pseudopod from a glial cell wrapped around the neurons. And at the intersection between the segments, things get kind of thin over here. And you actually get to the point where the axon becomes effectively directly exposed to the extracellular media, which is to say its resistance is lower over there. So what happens in neurons in order for them to conduct electricity fast is that an action potential forms at a node where it's possible to have the membrane currents be significant. And at those nodes, you'll get a high voltage potential over here. Because you have myelin around the neurons, that voltage drops off relatively slowly with distance over here. So when an action potential forms over here, it's able to recreate an action potential between the next two myelin processes at the next node. So actually, if you were to look at the voltage over here, you get an action potential here, nothing here, an action potential here, nothing here, and so on forth. And the myelin speeds all that process up. With MRI, it has, in the past few years, become possible to visualize perhaps the myelin. And this is a technology called diffusion imaging. We're going to hear significant lectures about that starting on Wednesday from here. But you can actually sort of trace out where the principal pathways are in the brain for this. The myelin, being made of fat, is relatively light colored compared to the neurons, which are made mostly of water, and that explains the difference between white and gray matter in the brain. The white matter is really just the fat in the myelin. And the connections across the brain are very dense through the corpus callosum and so on. Many of you, I'm sure all of you basically know that. One neuron communicates with another one by forming an action potential, but then the other neuron doesn't receive it exactly as an action potential. What happens instead is that small amounts of chemical transmitter go from one neuron to the other. And this was discovered and shown in great detail in 1956 by Boyd and Martin, who did this brilliant experiment where they depleted the extracellular medium of calcium. And when you do that, the little vesicles that leave the neurons don't form as well. And so you reduce the number of these events of little vesicles carrying transmitter to a vanishingly small number. And then they looked at traces of these individual events, the spontaneously occurring events, and they noticed that, oh, here's a tiny one, here's one twice its size, here's one 3 times its size, and they just counted how many times you had a small one, how many times you had an intermediate one, and how many you had a big one. And this is a histogram of the distribution of those events, which follows very closely Poisson's statistics shown in the red over here, all of which tells us that these are discrete events of essentially fixed size that can occur simultaneously or not. And so this was effectively the proof that you are moving tiny particles of neurotransmitter. This is way before electron microscopy was able to see these things. What do I want to show with this? I'm going to skip that slide because I don't remember exactly what I wanted to say with it. So if you then go ahead and study in great microstructural detail what's going on when one neuron attaches to the other, is you see these entities called synapses. And the synapse is a gap between the incoming neuron and the output neuron, or the receiving neuron for this. Those gaps are tiny, but there still is membrane on both sides of it, right? So the electrical current flowing from this neuron does not directly go into its receiving neuron. However, if you look in great detail in these gaps, what you'll see is, first of all, these little packets, little spherical vesicles, which are now understood to contain the neurotransmitter. Another thing you see in great profusion at the synapses are mitochondria. Does everybody know what mitochondria are? Maybe you do. Mitochondria are the energy maintaining apparatus and conversion apparatus in the neurons. The fact that you have a huge amount of mitochondria in the synapse is evidence. It's evidence that something going on at these synapses requires lots and lots and lots of energy. And then you also have this thing called the Golgi Complex, which most of you know is actually involved in the formation of vesicles. So this is a machine for creating these vesicles down there. Here's what they look like under scanning electron microscope. Under plain electron microscopy. You can see the vesicles. On one neuron, they're round. On the other neuron, they're sort of plate-like on here, like sickle cell-y kind of things. The different neurons, turns out, have different types of neurotransmitter in them. And it's generally thought that these sort of ovoid vesicles are containing inhibitory neurotransmitter and the circular ones are containing excitatory neurotransmitter, at least if you're talking GABA and acetylcholine, which is sort of an interesting effect. You can see the mitochondria, and you can see how a huge volume of the synapse is made up of mitochondria. And you can also see the membrane is continuous between these two neurons. Right? So they're quite separated. So the idea is an action potential comes down, and in the process of coming down, it facilitates the release of these vesicles, which then go through a small gap and reach and diffuse through that gap and reach the receiving neuron. And as a result of their going through that gap, they open pores in the receiving neuron, which then allow charge to go through and cause this receiving neuron to become depolarized and ultimately form an action potential. It takes a little time, a surprisingly long amount of time, given how fast we have to do things, about half a millisecond just to cross the synapse. And a great deal is known about this machine as well right now. And without really going too much into it, the important part, at least in terms of these older experiments and other experiments you might do as physiologists, is that this process all requires calcium. Without calcium, the fusion of the vesicles with the membrane doesn't occur. Each of the vesicles contains something we're going to call a neurotransmitter. We call it a neurotransmitter because it transmits information from one neuron to another, but almost any chemical in the body seems to be able to do that. It's really sort of an exaggeration, but there are an enormous number of things which act as neurotransmitters. The most common ones, by far, are acetylcholine and gamma-aminobutyric acid, or GABA. Acetylcholine is thought to be always excitatory. It causes depolarization of the receiving neuron. And GABA is thought to be inhibitory. It causes hyperpolarization. It moves the neurons away from firing action potentials. But evolution takes advantage of whatever chemicals are around to do whatever it can make them do. And so you see on the list, for example, of neurotransmitters, something called vasoactive intestinal polypeptide, or VIP. And it was called that because they found it in the intestines first before they figured out that it was a signaling mechanism within the brain. And many, many peptides have this effect. Okay. Now, in addition to these vesicle-based synapses, it's been known for quite some time that in insects there are something called electrical synapses, where in fact, rather than having chemicals which transmit, which carry stuff through the membrane, there are actually pores that allow the passage of current from one neuron to the next. This is what they look like in cross section, and this is what they look like in transverse section. And you can see the pores are these little circular things which have an opening in them. Electrical synapses are pretty interesting. For many years, people didn't think they took place in mammals or vertebrates, but in fact they do. There are electrical synapses in our brains, and they must do something different in terms of how they conduct things. One of them, of course, is they don't have a time gap between them. these things are actually protein channels which go from one cell to the other cell. And they form these very, very tight junctions. One of the things that's known about this, at least in insects, is they have the capability of moving current one direction only. So they act like like a diode, and would happen in the brain, which would mean that only one neuron can signal the other neuron, and not necessarily both directions. It's not clear that always happens. It's very hard to really know these. So electrical synapses have different energy requirements, of course, than the vesicular synapses, because you're not moving all this apparatus around, you're not creating neurotransmitter and so on for this. And so energetically, they're probably pretty close to silent. Another thing that is well known about the neurons is that a single vesicle transfer from one neuron to the other is not going to depolarize a neuron enough to make the receiving neuron fire an action potential. In fact, it's very unlikely that all the vesicles in a single synapse are likely to depolarize the receiving neuron enough to make an action potential. So instead, what happens is you have multiple synapses onto the receiving cell. They all have to occur essentially at the same time. The events have to occur essentially at the same time to cause an action potential to be transmitted. Each vesicle creates a single excitatory postsynaptic potential, or inhibitory, which lasts for, on the order of a millisecond, but is tiny. You have to move the neurons from about minus 75 millivolts to about minus 55 millivolts in order to get an action potential, and these things are only about 2 millivolts, so 10% as large as you would need. So for this to work, multiple EPSPs have to take place essentially at once. And by at once, I mean both they have to impact the same patch of the receiving neuron, because it's a local in here, because these have these spatial constants, but they also have to happen pretty much at the same point in time. They last long enough that if you have multiple events occurring just slightly offset, they tend to superpose. And the formal math to go from here to here is called convolution, and we'll be talking about convolution throughout the course. The dendrites on the receiving neurons tend to receive incoming information also in patches. So one set of inputs might affect this area, another set of inputs might affect this area, and so on. This is an efficient way of the neurons to aggregate information, so that different portions of the neuron might be receiving different but related information. It allows them to go ahead and form an action potential. Finally, as regards this stuff, if you look really, really closely at the dendrites, they're not just smooth surfaces, but they have these spines coming out of them. And the spines and these little red zones in here are where the actual synapses take place in these neurons. And you can see that the spines have a little bulb on them and a thin neck. Now, from the point of view of the electrical properties of this, what that does is create a high resistance to current flow through that neck, which means that an action potential here is going to depolarize this portion of it very, very effectively. So the spines become information receiving points which efficiently control how the incoming signal is carried to the output neuron. And one of the things that's now understood is that these spines come and go incredibly rapidly. Over the course of seconds to minutes, animals will regrow different spines. And this is one of the ways in which neurons adapt and learn information. Okay. How do neurons encode information since all of these action potentials are stereotyped? Well, this is an oscilloscope recording of a neuron, and during the time shown here in blue, it's receiving input. In this particular case, this is a visual system neuron, and the light is being turned on, and each of these vertical bars represents an action potential. And so here we'll just do it in real time. So what you're obviously hearing is that the neuron fires more action potentials during the time that stimulus is on. And so the rate at which this firing occurs is thought to be one of the principal ways in which the brain carries information through action potentials. Now, it could be multiple ways. First of all, the rates are somewhat limited because these action potentials are relatively, each individual one is relatively long. The neuron really under no circumstances is going to fire more than 1,000 of these per second, and more like hundreds per second when they're really maxed out. And so if you think about it, your range must be from up to 400 events a second to down to as low as what? Well, one action potential per minute would be pretty useless for our brains. Right? So there's a lower range in which these things have to be firing enough for us to be carrying information. So the dynamic range must be on the order of a few per second to a few hundred per second, and that's all, right? They just can't convey much more density of information for that. The implication is that something about the brain is able to measure differences in rate with incredibly high precision. If you think about with our eyes, we can see many orders of magnitude in different light intensity by only varying the rate of these neurons firing on the order of magnitude or two. Right? It's a remarkable thing. Time in the brain is a big old mystery, big old mystery. The other thing that can happen, of course, is that in addition to the firing rate of these things being a signal mechanism, each of these axons comes from someplace else. And it acts as a sort of a labeled carrier. The thing from your ear, from your cochlea, goes to a particular part of the brain, and anything that comes from the cochlea is probably auditory information. Neurons can change how effectively they allow the transmission to take place by modifying the spines, for example. That's another way your brain can change the information content. And of course, the neurons, these action potentials have to be semi-synchronous for them to do information transfer. So another way your brain can control information is the synchronicity of these events. Here's a simple example of this idea of labeled lines in place encoding. This is a schematic of the inside of your ear in the cochlea. And in the cochlea, it's known that low frequencies vibrate one portion of the basilar membrane, and high frequencies vibrate a different portion of it. And so if you have sort of a middle range frequency over here, only the middle portion of the basilar membrane moves. And so only the neurons which are innervating that portion of the basilar membrane fire signals, and therefore you can infer the frequency of the sound you're listening to based on which of these neurons fire. I mentioned this notion of inhibition. It's also possible with some transmitters to hyperpolarize the cell, move it further away from firing, and therefore they control the extent to which that neuron's going to respond to other positive going inputs for this. And the inhibitory stuff does typically require the transfer of a neurotransmitter. However, it doesn't involve the motion of sodium. It involves mostly the motion of chloride, for which there is not an active pump. So let me try and make that clear. So the chloride concentration follows the sodium-potassium concentrations for this. So producing inhibition like this does not require the pump necessarily to run more rapidly. One of the effects is that we, to this day, don't know whether inhibitory synapses are going to show up as an energetically detectable signal in things like bold imaging. We know that excitatory things do. We don't really know about inhibition. And when you see so-called negative bold effects, it's very tempting to go ahead and say, oh, well, that's an inhibitory process. You have no evidence of that, not yet. One of the interesting things about inhibitory synapses is that the majority of them in the brain are what are called presynaptic inhibitory synapses. That is, I have an incoming synapse positive, this gold one over here, and right before the signal reaches the synapse, there's an inhibitory synapse impinging on it. And effectively, if this is inhibiting this guy from firing, it gates that transmission. This was discovered first in the spinal cord, where it's part of a very, very well-known reflex process, but it is ubiquitous throughout the brain. Okay, so given all of this, what kind of things might we be able to detect using functional imaging methods or imaging methods to detect presumably is the energetics, presumably driven by things like the sodium-potassium pump and the cycling of transmitters. We can detect direct electrical signaling if you happen to be able to get an electrode near the neurons. That's kind of tough in humans, but it is done routinely now, and research is done that way. You can also measure the so-called electroencephalogram, which is the distant voltage effects from neurons inside the brain, much attenuated by the whole idea of having a skull in the distance and also blurred by those things, but you can detect it from the outside. If you have high enough resolution, you can detect differences in shapes of these neurons, which gives you some idea about their functional differences. You can also do chemical measurements from this, either direct sampling or now using a tool like magnetic resonance spectroscopy. We're not going to talk about spectroscopy in this course, but it is a window for looking at chemical concentration. There are other factors which give evidence, like how many neurons there are per cubic millimeter, and you can detect that. You could get a reasonable job at that using spectroscopy and looking at a particular chemical which is specific to neurons called N-acetyl aspartate. And then the MR image really shows a lot of difference between fat and water, which is part of the reason we see this huge gray-white contrast in the brains into the MRI. And so that gives us evidence of where the pathways are. So just briefly, and we're going to have Nikos Logothetis, who is the perfect master of this topic, come and talk to you guys about energy and neural firing. But just to get a couple of high points from here, the presynaptic neuron requires, in order for it to work, transmitter synthesis. Every time the transmitter leaves the cell, it has to be reformed, and that's an energetically demanding thing. The process of actually forming fusion with the cell membrane also requires some energy. And then in addition, this chemical which goes out into the extracellular milieu has to be scavenged for two reasons. One is that it's toxic, and the second is that it can be reused by these neurons. And so there's an active mechanism of reuptake of transmitter in the presynaptic neuron. In the postsynaptic neuron, every time an action potential forms, the battery runs down. And so you've got to drive this sodium-potassium pump. We don't really know about the inhibitory costs of these neural firings, as I mentioned, but there's some good experiments to be done. Another feature of mammalian brains is this notion of a cortical column. And this is now a very close-up view of a nose of a baby mouse. And here is little whiskers. They have a small number of whiskers that are used for sensing their environment on the order of 40 to 50 on each side. And they occur in a regular pattern from this, just like the 5 fingers on your hands occur in a regular pattern. And if you go ahead and stain a portion of the brain here on the sort of dorsolateral surface, you'll find that that you see this sort of grid pattern to certain kinds of stains. And it's known that the centers of these grids are mapped to single whiskers on the outside of the animal's nose. And all of the neurons, if you poke an electrode in here, fire in response to motion of the associated whisker. So for example, I move whisker B, and the neurons in this region all respond to this. However, you move just a little bit to the side, and they respond to a different whisker. And so in a sort of columnar way through the cortex of the brain, you see these organized functions. So that's known throughout mammalian systems. We have cortical columns associated with different places in our visual field. We have cortical columns associated with different portions of our skin and so on for this. But it's a key organizational feature of the brain. And the cortical columns themselves are on the order of a couple of hundred microns in the human brain. When we do functional MR imaging, our resolution is relatively crude compared to the cortical columns. These days, in our lab, we're scanning usually 2 millimeter isotropic voxels, which therefore contain several hundreds of cortical columns within it. And since they all have different function, we don't really get to resolve that very, very well. However, there are very high resolution techniques that people have used which seem to demonstrate that with functional MRI, you will have separable excitation of the columns. And given that functional MRI, as you probably know, and I'll be describing in a minute, is following blood flow, that implies that each of these cortical control of blood flow. And of course, each column contains thousands of neurons. We don't see single neurons with any of these tools. Why did I want to show you this? Okay, so again, our pictures of the types of neurons. I said that the pyramidal cells were ubiquitous in cortex. One of the things that's interesting about them is that the axon will be on the inner surface of the cortex, and the dendrites will be on the outer surface of the cortex. So they're highly polar cells, and they're all oriented the same way. They're all oriented normal to the surface of the cortex. A consequence of that is that here's our sort of surface of the brain and the outside of the skin. A consequence of that is that when the neurons fire action potentials, they generate a current from the cell body to the dendrites, or actually, sorry, when they're receiving input, a current from the cell body to the dendrites. However, if you have thousands and thousands of these neurons all oriented the same way, the currents are all going the same direction. And with enough of this going on, you end up with effectively a single relatively large current going through these hundreds of neurons, which is reflected as a voltage potential difference at the head. The current, the charge separation in these currents generates a voltage difference. And that voltage difference is what can be used for the electroencephalogram. It's the summated effects of many, many neurons. The EEG is problematic in so much as if you have two sets of neurons which are on opposite sides of a cortical fold, they're going to be self-canceling at the surface, which is sort of a drag. There are some other issues with EEG. For example, the signal falls off very, very rapidly with distance, falls 1 over the square of the distance. The magnitude depends on the orientation of these electrical dipoles compared to the surface. The temporal synchrony is required. We can't detect a single neuron firing from here. The neurons have to be firing in the same sort of direction at each time. Nevertheless, you can pick up a macroscopic signal. This is 30 or so electrodes on different parts of the surface of the head. And one of the prominent things you can see in here is these large sinusoidal oscillations in here. Look, this is the heartbeat, right? So you get some sense of time scale. These oscillations are occurring at about 15 times a second, 10 to 15 times a second. Does anybody know what that is? What's that called? Really? Hertz. Oh, hertz. Yeah, times per second. Okay, I'm sorry. What are these oscillations called? Alpha, yeah, alpha. Alpha is this crazy prominent thing that in brains of all mammals, you stick an electrode on their head and you start seeing these spontaneous oscillations of currently unknown function. However, just to sort of bring this full circle with this, you can measure it at the same time you do MRI. So here are some spontaneous fluctuations in alpha power in an individual. You just leave them alone and the alpha power goes up and down. You can model what the expected blood flow response is, and I'll talk about that in a minute, which might look like this red curve. And then you can find portions of the brain which mimic the power in the alpha band. And you can see now that, in fact, there are regions of the brain which very specifically increase signal intensity when alpha goes up or decrease signal intensity when alpha goes up. I show you this in part because I find it fascinating, but in part because it's also sort of linking this idea that the local electrical properties of the neurons can be proven to be associated with the bold response. I'm going to skip this just because I'm taking too long. Okay, so final slide in this section. There are many, many features of neurophysiology which become visible using magnetic resonance. And those include the energetic demands. And the energetic demands from these various components that I've talked about become visible through blood flow effects using technologies such as bold blood oxygenation level dependent signaling, arterial spin labeling, which is a flow effect. We can, I'm sorry, these are all lined up strangely from this. There is the capability now under certain circumstances to actually image the sodium concentration. Sodium turns out to be one of the nuclei that MR can pick up. It requires relatively exotic hardware and technology, and it's a relatively low resolution, but people have done sodium imaging. And the sodium imaging might be a window into the energetics of the brain. I mentioned spectroscopy. This is a form of chemical analysis which you can look at the chemical concentrations in individual voxels in the brain and therefore measure things like glucose concentration or NAA concentration. There is a literature about the idea that you should be able to image the electrical currents in axons. I'm a skeptic about this. It doesn't mean these people are wrong, but I'm a skeptic because, as I said, the current through axons doesn't look like current through a wire, and you can't model it as current through a wire. But most of the models that we use in MR imaging of currents assume effectively that you have a constant current flow through the axons. Just not true. You can look at the diffusion of water molecules. We'll talk about that later using techniques such as diffusion tractography. And finally, you can look at, for example, the concentration of chemicals which are local to different cell types, and you get a map of the distribution of those cell types. Catch my breath, and I'm going to move on to this topic of a brief intro to bold imaging, and let's see, I'm supposed to go to 10:30, so this will be quick. Any quick questions you've got here? Okay, so in a couple of slides, I want to orient you to the basic bold experiment. And I want to tell you a little bit about the course design this year, which is a little different than in prior years. In prior years, we spent a relatively heavy amount of emphasis on MR physics. This year, we've mostly removed that, at least the basic physics portions of this. And that's based on the fact that most of you people claim to have a substantial amount of MRI experience. And so I'm not going to talk much about it in this slides. However, you'll see in the schedule that I have a sort of optional periods of MR physics. I love to talk about MR physics. Do it all day long. And I will do that for you. And you don't have to be embarrassed to be in that portion of the course. But if you feel like you know everything, you're in good shape, you can skip that. Okay. Functional imaging, functional brain studies on the basis of blood flow. This is a really cool slide. This is a fellow named Angelo Mosso, who in 1884 built a little platform that's a balance. It's got a little a bed on which somebody lies and a little edge on which they can float it, and a set of apparatus to make things balance at the center of the body. Okay? And then attached to the end of this table is a pen, which is writing on a little rotating disc. And if the balance goes down towards the head, the pen goes up, right? Simple thing. He did these experiments on people who had open skull wounds, okay? Which meant that the volume of the brain could change because there's space for the brain to go. And what he found, amazingly enough, was that if people did mathematical calculations in their head, the balance tipped downward on the head end. From which he inferred that blood flow increased to the brain when people were doing mental calculations, right? That was the first useful functional imaging experiment, as far as I'm concerned. A brilliant, brilliant thing. And he ends up quoted by many of his followers, one of whom, the famous William James, you can't say William James without saying the father of psychology, who says, "We must suppose a very delicate adjustment whereby the circulation follows the needs of cerebral activity. Blood very likely may rush to each region of the cortex according as it is most active. And of this we know nothing." So he is already aware that blood goes into the head. But he came up with the idea that maybe it depends on what part of the brain you're looking at. which is stuff he would have gotten from listening to people like Paul Broca, who had already shown that different parts of the brain do different things. The notion that you could study blood flow effects was very exciting, but it took another 100 years before somebody figured out the first way to do that. And that would have been Michael Phelps and John Mazziata at this institution, who showed using PET imaging, positron emission tomography, that the concentration of energy-containing compounds, particularly in this case glucose, changed in various parts of the brain as you use them more. And this is a difference image between people looking at a blank room and watching cars go by on Westwood Plaza, and you see this large activity, for example, in the visual cortex from this brilliant experiment. The first MR versions of this were done by Jack Beliveau. Jack Beliveau tragically died 2 years ago. Jack Beliveau was a postdoc working at Harvard, we had built this ultra-high-speed scanner, and Jack got the idea that you should be able to watch blood flow in the brain using this high-speed scanner. And so what he did was arrange to inject a chemical into the arms of people, a gadolinium contrast agent, and when the chemical went through the brain went dark. And if you did this in darkness, in this blue area, you get this brief but relatively large signal change, 20% signal change or more, or in this case 25% signal change as the thing goes through the brain. On the other hand, if you do this in the presence of blinking lights to the eyes and you're measuring the signal in the visual cortex, what happens is you have a much larger signal change. And the difference between those two became evidence of activation in the brain. These were the first functional MRI experiments. Anyway, it was on the cover of a well-known magazine showing the regions whose signal changed. Now, I talked about Mosso's balance. Here's another balance. This is the balance owned by Linus Pauling. It presently sits inside the president's office at Caltech. And an interesting thing about this balance is that Pauling, who is a chemist, realized that when oxygen binds to hemoglobin and iron, it has the possibility of disturbing the spins of the electrons in the iron, a consequence of which is you change the magnetic properties of the iron. And so what he did was put blood in one of these balances, balance it all out nicely, and put a magnet beneath it. And lo and behold, the blood, when it was oxygenated, got pulled towards the magnet, whereas the deoxygenated blood did not. Phenomenal experiment. Phenomenal experiment done with, household apparatus. Pauling also got a Nobel Prize for this and a great deal of other chemical work. So like Mosso, it's understood, and if you think about the physics of it, it's well understood that that if you have a magnetizable object inside the head, it will cause a signal change in the MRI, particularly if the magnetizable object is inside the blood vessels, it causes the signal to decrease. That's the basis of the Bold effect. And the changes in magnetic field with distance are very, very large when you're near a capillary, and it gives relatively large magnetic field disturbances and therefore large signal disturbances. The effect is to shorten the duration for which the signal is present in the MRI, the so-called T2 decay. And this is work done by Keith Tholborn in 1982, and this was another absolutely brilliant experiment. He realized from the Pauling studies that it might be possible to see this stuff using MRI. And in this particular case, magnetic resonance is kind of pre-imaging days. And he measured the rate at which the signal decays as a function of blood oxygen concentration. And you get a perfect straight line. Beautiful, beautiful experiment showing a direct effect of blood oxygen on the magnetizability of blood. On that basis, two groups, Seiji Ogawa's group at Bell Labs and Ken Kwong's group at Harvard, got the idea that you should be able to use MRI to look now at the signal changes associated with blood oxygen. And these are Ogawa's work from this, showing the blood vessels in a deoxygenated condition showing up dark, and in a hyperoxygenated condition showing up relatively bright. And the difference between those two is showing where the blood is, where the deoxygenated blood is. These are Ken Kwong's original human data from these experiments. This is the back of somebody's head, using a little receiver coil on the back of the head, and in the presence of blinking lights. And this is a movie of those primary data, and you can see the signal fluctuating a little bit with noise, right? If you squint, you might be able to see that this region of the ear is dark here, and then goes bright, and then goes dark again, okay? But that's easier to see if you simply remove the mean signal. And that's what's done over here. And so you can see dark and bright fluctuations in here. This is the raw data in a good, robust MRI experiment. The very first experiments worked. Phenomenal thing. When Ken first did this, I've got to say that the other people who worked in the lab, me and everybody else, didn't believe it. Artifact, cool artifact. But in fact, he was proven right. And so in a good day, a bold experiment has such high signal-to-noise ratio that you can see the effect by eye, right? And demeaning seems like a pretty easy transformation on the data, right? And you can see it even, I mean, it's like obvious from here. So the intensity variations are known to reflect differences in blood oxygen. However, the signal goes up when you're in presence of stimulation, when you have presumably more oxygen being consumed, therefore lower MR signal because you have more deoxyhemoglobin. So why does it go up? That's sort of a paradox. And the guy who principally worked this out, I should have credited him on this slide. Oh, I can't blank his name right now. That's too embarrassing. Bob Balaban, came up with this. Oh, no, I'm sorry, I apologize. Rick Buxton came up with this theory that, and it goes like this. This is a capillary shown in gross magnification. Red blood comes in, blue blood goes out, and the reason it turns blue is oxygen flows out from the neurons into the brain. Out from the blood vessels into the brain, and it simply follows its concentration gradient. The capillaries are so thin that the oxygen simply diffuses passively across the capillaries, and it delivers enough oxygen so that the brain is kept at a relatively high oxygen concentration. Okay. Now, when the brain work increases, the oxygen content in the brain goes down. Therefore, there's a higher gradient between inside and outside of the capillaries. Therefore, more oxygen flows through. And as a consequence, what happens is that the blood turns blue more quickly because most of the oxygen has flowed out in the initial segment of the capillaries. And that starts starving the brain again of oxygen because it's all gone from the capillaries. So the response in our brain is to increase the flow of blood through these capillaries. So as you increase the blood flow, more red blood cells here you can see, what happens is that the oxygen, there is still oxygen in the capillaries by the time you get to the distal end of the capillaries. Okay? However, what's happened now is that you're delivering both high oxygen concentration by the distal ends of the capillaries, but also at high flow rates, which means now the distal end of the capillaries, now when you reach the venules, are now hyperoxygenated compared to the normal state. And the result is that you end up with an increased MR signal because, in fact, you have not more deoxyhemoglobin in the brain, but less. Okay? So that's how this happens. So when the concentration gradient is maintained throughout the capillary beds, more oxygen is delivered to the brain, but also more oxygen appears in the venules. These are, again, Ken's raw data. And here you can see, just looking at the time course in a few voxels, what happens to the signal intensity. And you saw this by eye, so it's no surprise that the signal changes are huge. In this case, a few percent. You turn the stimulus on, the signal goes up. You turn it off, the signal goes down. But none of this is actually instantaneous. And if you look at this, you can see that it sort of takes a little time to rise. I'll talk about that in a moment. There are other properties you can measure. For example, the T1 relaxation rate. Again, if we talk about physics later, you'll see this. But the T1 is sensitive not to blood oxygen concentration so much as to blood flow. And you see similar visual maps when you're measuring the T1 effects as to the T2 effects. Ogawa concentrated on the T2 effects. Ken showed both the T1 and the T2 effects in the same time, but they're very prominent. This is the basis of another form of functional imaging, which is just a flow-dependent imaging. People don't do it as much. It's slower, and its resolution is not the same. The first human brain mapping experiments, where you're going to show differential function in here, were relatively simple. You shine light in the right visual hemifield or the left visual hemifield, and you activate the contralateral portions of the brain. So here, light is turned on the, let's see, this is the left hemisphere, it's turned on the right visual field, signal goes up. If you switch the sides, you get the inversion from this. You get a very crude map of visual fields in the brain. You can look at more nuanced things. I think that this was the first experiment that truly got me excited. I mean, I participated in the original ones. I shouldn't say this is the first one that got me excited as a neuroscientist, saying we're learning something new about the brain. This is Roger Tutel's work. Roger was interested in the motion-sensitive region of the brain in monkeys called MT, which in humans is called V5. And he simply showed humans moving pictures, gratings that actually move. And he saw not only the primary visual area, but also this more lateral portion of it, which is the presumptive motion-sensitive region of the brain. Well, how did he make that presumption? What he did was study the response in that region as a function of the different stimuli. So here, we're looking at moving gratings of either low contrast or very high contrast. And in the primary visual area, you can see the signal increases monotonically as the contrast increases. In this MT region of it, the signal is constant response in the brain, essentially regardless of contrast. On the other hand, if you alternate between moving and stationary stimuli, you'll see that V1 doesn't care whether these things are moving, whereas MT, or V5, responds only if they're moving. So you actually did a functional identification of that patch of cortex. Very cool experiment. So to just sort of close up this section of here, the typical traditional form of MRI analysis is to present a task and look at the observed signal change in the brain. And if the signal changes are robust enough, like in the early visual areas, you see these large signal changes associated with the task timing. In practice, however, the signal changes are usually somewhat weaker, and they have this non-instantaneous time course. So it's more efficient to model the observed signal from first principles and then search in the brain for regions of the brain that respond like your model, right? Because your task timing is not the same as the signal response in the brain. So you want to model the time course. And the way we developed to do this was to look at what would happen if you were to just make a brief pulse of light into the eyes. And you get a time course for the bold signal which lasts several seconds and itself is much longer than the light pulse. This happens with 1/10 of a second light pulse, and yet the response is sustained for 20 seconds. But if this is the response to an instantaneous flash of light, then it turns out through basic linear systems analysis, you can predict the response of the brain to light pulses of any duration or shape. So this is now, in green, the predicted model of the brain response if we take the task model and mathematically convolve it with the impulse response model. And the convolution of those two looks like this green course over here, which you can see models incredibly well what's happening in the brain. And here, just another observation of it, the stimulus on and off, the bold signal time course, which is delayed with respect to the stimulus, and the modeled response from here. So basically all the analytic tools you use these days do some sort of convolution model in order to improve their sensitivity. This is a fun observation, and I harp on this constantly, even though this is 20-year-old work. If you change the amplitude of the stimulus, you get a scaled amplitude of the response in the bold signal. However, it's not a linear scale. In this case, we had people tapping their fingers either once per second or up to 4 times per second. And what you see in yellow are the observed responses in the brain to these different finger tapping rates, and you can see that it increases as the finger tapping rate is increased. However, these are going in log units, 1, 2, 4, and the signal intensity is not going up 4 times as much when you're getting 4 times the stimulus. Nevertheless, if you model the amplitude response as the logarithm of the signal intensity, as shown here in this blue or purple or whatever it is, it's a very, very close model to the brain. Same things happens in the visual system. You have a log normal kinds of response. All right, with that, I have a couple of minutes to answer questions about bold and functional imaging and whatever, and if there are any, here I am. What are we going to do about this? Okay, well, I'm assuming that you guys are going to start asking questions soon enough and that I just gave you the clearest presentation you ever heard, or that you didn't understand a word I was saying. And if that's the case, come up to me after. Yeah, take the mic.
Well, I guess my question's more about the different kinds of convolution models.
Mm-hmm.
I mean, we have like some basic ones we use that are kind of available for us, but there's also some others that are out there that are kind of like custom convolution models, and
Mm-hmm.
I'm just wondering how you get those. What are some trade-offs? For example, I'm thinking about physiological signals, like peripheral physiological signals, and you
Mm-hmm.
might use some alternative convolution models rather than like double gamma.
Okay, yeah, so I was hoping to bring up a particular slide, which I'm not finding right away. Where did this come from, this particular shape, okay, that we're seeing here? And the answer is Bob Savoy, and I've forgotten her name now, Kathleen, one of his students. I'm embarrassed because this is going out in public. The two of them did a simple experiment in which they flashed a brief pulse of light in somebody's eyes and observed the response. And they did this many, many, many times and averaged them. And you get a shape. And so in order to come up with this model of the hemodynamic response function, what I did was fit their observed data to a particular low-dimensional equation called a gamma-variate function. Low-dimensional meaning there's only three parameters to fit it, and one of them is amplitude. The other two are time. And it's parsimonious to fit things with the minimum number of variables for this, right? Because the fits don't become, you don't have to correct for the number of parameters in there when you're trying to measure the models. And that particular model was done on the visual cortex specifically. However, there's no a priori reason to believe that every part of the brain is going to respond the same. In fact, the visual cortex has at least from the level of the macrovasculature, a really different architecture than other parts of the brain, that, for example, the hippocampus. Nevertheless, this model has proven to be extremely accurate in most cases. Now, if we go and look back at Ken's data, I'm sorry, quickly get there. This is again as a visual cortex stimulation. Turn the stimulation on, signal goes up, turn it off, it goes down, but it goes down below baseline. And that going down below baseline is not captured in that single unipolar hemodynamic response function. So other people, and I think Gary Glover is the one who started on this, suggested that you could add 2 more parameters to this thing, 3 more parameters, excuse me, and add another gamma variant function, which is slower and goes the opposite direction, so that the response goes up, goes undershoot, and recovers. And that's the so-called double gamma model for this. Formally speaking, the double gamma model should always fit better because you've got 3 more parameters to fit, okay? And that's cheating, right? Cheating. If, on the other hand, you fix those 3 parameters, essentially in ratio to the single gamma model, then you're not cheating the same way, right? It's okay. Then it's a question of which model actually fits the data better. And this is not a trivial question, because the signal-to-noise ratio in bold experiments in places other than the visual cortex is not typically this high. And so the different models will fit differently based on noise, which is always going to happen. And so whether to use one model or the other is a little unclear. I have on my computer, and maybe in the break I'll quickly come up with which slide it is, showing you portions of the brain which have an undershoot prominently and portions which do not have an undershoot prominently. In fact, the visual system has the most prominent undershoot of any part of the brain that I know of, whereas motor cortex has virtually none. So again, which model to choose? It's very tough to find a guideline. You know, you would probably be well served to do some sort of an impulse model, the equivalent of a flashing light, in whatever specific brain region you're studying, and average and average and average and average until you get a good idea of the shape, and then fit that. That's easy to do in the visual system. It's relatively easy to do in sensory motor cortex. It's really pretty hard to do in mathematical reasoning cortex, right? To have a little impulse of, oh, damn, that's an integral, quick. And so coming up with a general model for this is very, very difficult. It would be kind of nice if we could do direct electrical stimulation onto different parts of the brain and see this, but I don't see that in the offing anytime soon. The Glover double gamma model, the way it's typically shown, is actually kept this low dimensionality. You fix the ratios as these time constants. On the other hand, the various software analysis tools, for example, FSL, allows you an extremely flexible fit for this. And really what you should be doing is correcting your p-values by the number of free parameters you've allowed in that fit, right? Generally speaking, you have to go ahead and say that you have more degrees of freedom and therefore less statistical significance for any given quality of fit for these data. So that's, I mean, it's a long answer to a simple question, but that's where I am with that. Mike? I think you just turned it off.
Hello.
Oh, you're good. Okay.
So I have 2 questions. The first question is about inhibition. So in one of the earlier slides, you mentioned about negative potential that does not result in action potential.
Right.
So it's just not possible, or it's not known, kind of.
I'm sorry. The inhibition is an electrical effect. It's extremely well characterized, and it drives neurons away from action potentials. That's not controversial. What is not known is what that does to the blood flow response.
The second question I have is about myelin sheaths and the speed of response. So it seems that it's directly proportional. If your myelin sheaths are shorter, your signals will move faster across the neuron?
That's interesting. Actually, I would say in general that would be inverse to that. That is the formation of the action potential at adjacent nodes happens when the voltage in one node is high enough to increase the voltage at another node sufficient to cause transmission. Okay? If you had short internodal spacing, it's an implication, really, that the voltage is dropping rapidly between these nodes. And each action potential happens at a different time. This one, this one, this one, and this one. And I don't know this for a fact, so I'm only sort of hand-waving and guessing over here. But I believe what you would find is that if you studied this in real nerves, that the larger internodal spacing was associated with faster neurons and thicker myelin. Yep. It's good. We passed the mic. You don't get to hear the
I was wondering if you could talk a little bit about your opinion about directly measuring physiological dynamics in the scanner, like cardiac activity or breathing rate, to kind of model those artifacts in your signal processing, or if it's not really necessary to do that?
So we will have formal talks on that, which is a relatively broad topic. However, there are many good reasons to measure these things because they do have effect on the signal. And to a certain extent, regressing them out means you have a better model. But some of these are perfect confounds, right? So in that original Masso balance experiment, he had to correct for that. Because as more blood flow comes in with faster heart rates, that also had to be adjusted for. Anything else? Yep.
So I know the negative volt effect is like kind of difficult to interpret, but what do you do when you see these negative volt results? You just go away from them, or?
My inference is that if I'm comparing a negative bold signal to baseline, that region of the brain is is seeing less firing of the neurons. Whether it actually is as a consequence of inhibition through GABAergic cells, I can't tell. I can tell there's a cool experiment to be done. I've been wanting to do this for the longest time. You can actually inject chemicals that facilitate GABAergic transmission in the brain. You would then go and stick it into the internal carotid on one side of an animal and compare it to the other side and see what happens, right? Because you get specific GABAergic effects when you do that. But so far, I haven't been able to pull that off. It's a relatively simple experiment. Another thing you might do is associate the electrical activity in the dentate gyrus of the cerebellum, which is known to be 100% inhibitory in its outputs, with neuronal activity. Cool experiments. Just put my name on the paper. Others? Okay, so let's take a short break. There's a little bit of food back there. You can stretch. The restrooms are immediately to the left of the door in the room over here. I guess we're good. Thanks.